enzymes are proteins that are catalysts for reactions in biological systems. What do I mean by catalysts? What does a catalyst mean?
They start a reaction.
Okay, so they start it. What else do they do?
Speed it up.
Speed it up. Good. So an enzyme is basically speeding up the rate of a reaction. It's going to make that reaction go faster. It increases the rate of chemical reactions. So they function by increasing the activation energy for a specific enzyme reaction. What is the activation energy? Because there should be a review. What is the activation energy? What is your activation energy? The activation energy of a reaction. Takers. Come on, take a guess.
With the amount of energy that needs to be used for that being required.
Okay, so the amount of energy that is required or that needs to be used, well said, to achieve that chemical reaction. The minimum amount of energy. Now, during the rate of a reaction, you have an enzyme binding to a reactant. That reactant we call a substrate. So whatever is hanging around that the enzyme is going to interact with is called a substrate. So that enzyme binds to that substrate. We get an enzyme substrate complex, which is illustrated here with ES. And that leads us to a product, some sort of end result or some sort of product in the end, and then the enzyme can be spit back out. Okay, now what's important to understand here is that these equilibrium arrows indicate that this reaction can take place in the forward direction, and then it can take place in the reverse direction as well. All right, so we have our enzyme plus the substrate, gives us an enzyme substrate complex, and finally that is turned into some sort of product or end result, and then the enzyme is spit back out at the end. Now, what do you think is going to determine the direction of this reaction? What is going to be a determining factor in whether this goes forward or whether it goes in the reverse direction?
If we need more energy or not, depending on what we're breaking down, the enzymes breaking down.
Okay, so energy you said?
Yeah, so it's like depending on the amount of energy that we need. If it needs more energy or if it can take away energy, like
Okay.
somewhere else.
Okay, yes. So what is going to determine the energy then? What's going to determine what needs more or what needs less energy?
That's being broken down. Maybe the product that's being broken down.
Yes, so the product. What about the product? What about the product? It has to do with the product and the substrate. What about them will determine whether this reaction goes in the forward direction or whether it goes in the reverse direction? Right, whether our body needs it or not. Right? So if we have lots of product, do you think this reaction is going to go in the forward direction? Are we going to make more product? No, we won't, right? We have lots of product. We're going to start making substrate again. So the reaction is going to proceed in the reverse direction. If we have lots of substrate, alternatively, the reaction will proceed in the forward direction and we'll make more product. What sort of general law does that tie into that we looked at before? What did I tell you was the right, negative feedback? Good. So that sort of ties into negative feedback. You have a lot of something, you want to turn it off, you sort of make a signal to stop that, and that's negative feedback. That's another theme that we'll see throughout this course, is that not only is there form following function, but there is efficiency. The body tries to maintain a certain amount of efficiency. And there's no efficiency in making more product if you already have lots. All right, so let's look at the activation energy barrier. Any questions so far? Let's look at the activation energy barrier here. Okay, and this is basically how the reaction proceeds in the presence and absence of an enzyme. All right, so we got, we'll put time on the X here. I'll explain how that differs in a little bit. And then we'll put energy. Okay, now we start off with some reactants. And then we're going to end off with some product. Okay. Now, in the presence of an enzyme, we'll draw our first slope here. And then we'll draw another slope indicating the absence of the enzyme, right? How the reaction proceeds in the absence of the enzyme. Reactants, sorry. Yeah. This is reactants. So we got reactants, we got product, all right? And this slope in red, let's make a key here, is without enzyme. And in blue is with the enzyme. Okay. Now what we'll see is that the activation energy, what did we describe the activation energy as? The amount of energy that's required for the reaction to proceed, right? The activation energy in the absence of the enzyme is going to be much higher. Let's draw that in here. This is the activation energy. Okay. Versus the activation energy, let's try to make a little here. In the presence of that enzyme. Okay, so we can see that the enzyme does what to the activation energy? What does the enzyme do to the activation energy for the reaction? Come on, just run through this. What is the, It lowers it, thank you. So the enzyme lowers the activation energy of this chemical reaction. Right? Now, what else can we say about the time in the presence and absence of the enzyme? Just looking at this graph, what else can we say about the time?
It takes less time.
It takes less time, perfect. So the amount of time that it took for this reaction to occur in the presence of the enzyme is significantly less than the amount of time it took, let's draw this here, in the absence of the enzyme. Okay, so I'll say this is enzyme versus no enzyme. So as a result, we can see that the enzyme does two things. It lowers the activation energy of the reaction and it speeds up the reaction. It makes it go faster. Alrighty. Anyone still working on this? Can we go?
Can you say the two things that the enzyme does again? It lowers activation energy and what?
Right, so it lowers the activation energy, or the amount of energy required, as well as it speeds up the reaction. So it makes it go faster. It takes less time. Okay. Now this is a similar graph to what we just drew, with the exception that on the x-axis here is the reaction coordinate. So this is not necessarily time, but what we can see here is the change in the activation energy. Alrighty, so we talked about an enzyme. We said that they do two things: speed up the rate of the reaction and decrease the activation energy. Now, what is preventing enzymes from just catalyzing reactions haphazardly in the body? Right? What happens to an enzyme? What features of an enzyme prevent An enzyme is catalyzing things left and right in your body. What is it about an enzyme?
Whether it's activated or not.
Okay, whether it's activated or not, and what determines whether it's activated? Can it just be activated?
Certain protein.
Okay, certain proteins that have what? Receptors. Good, so there's a specificity, right? So these proteins are unique in that only one type of substrate can interact with this enzyme. Okay, where does it interact? What? The active site. Perfect. So there's an area on the enzyme where that substrate can interact, and that is called the active site. And so the substrate specificity is this idea that each enzyme can only interact with one type of substrate. So there are two models that we're going to look at. The first model is called the lock and key model. Like the lock and key model. And this basically is what we're illustrating here in this image. We see an enzyme, which is enzyme E here. It has an active site. Now the configuration of this active site is unique in that only one type of substrate, in this scenario substrate C, will actually fit into that active site. So we call this the lock and key model, and this is just one of the theories that has been proposed as to why enzymes are specific. Okay, so there's a unique shape to that enzyme's active site, such that only one type or one shape of substrate can bind there. Right? Substrate A will not be able to bind there, substrate B will not be able to bind there, and so on and so forth. The second model is called the induced fit model. The induced fit model. Well, actually, let's track back a little bit. One of the drawbacks of the lock and key model is it does not explain the reversibility of enzyme reactions. What does that mean? What does that mean? So the lock and key model says that one unique shape of a substrate will bind to an enzyme's active site and sort of unlock it like a key to a lock, right? It does not tell us how we can go from getting product back to that uniquely shaped substrate. So it does not explain the reversibility of most enzyme reactions, right? So that's a drawback or a limitation of that model. The second model is the induced fit model. This one's a little bit different. We have a substrate that can interact with an enzyme, and as it gets close to the active site, it then changes the configuration of the active site such that it can bind to that enzyme and interact with it. Right, so we have substrate S in this scenario. Okay. The active site on this enzyme has this sort of square-shaped configuration, and when substrate S gets in contact, it changes the shape of this active site so that it can bind or interact, and then we have the reaction proceed, and we get the product. The enzyme is then spit back out with the product. So we basically have, question?
So the substrate is causing a change in the enzyme shape, or is the enzyme changing for the substrate?
The enzyme is changing its active site to accommodate that substrate. Okay. And then we have the product and the enzyme that is spit out at the end. Any other questions? Any other questions? And again, these are two theories, right, or two hypotheses that were proposed to explain why enzymes are so specific and why they're not catalyzing reactions haphazardly in the body. Okay, let's move on to talk about cofactors and coenzymes. Two important additional components to enzymes that help the reaction to proceed, but they're very different from one another. Right? Cofactors are non-protein components of some enzymes, and they are necessary for them to hold their function or their shape. Okay, these are usually trace metals such as magnesium, copper, zinc, and the unique property, what differentiates or sets aside cofactors from coenzymes, is that they are a part of the catalytic activity of an enzyme. Okay? I'll give you an example. We have, again, DNA polymerase. we have a strand of DNA. Now, DNA is very negatively charged, right? So we got lots of negative charge around DNA. And we have magnesium. Magnesium is positively charged. And so what magnesium does is it comes in, it's going to assist DNA polymerase in its activity by stabilizing the charge of that DNA strand. Okay, so magnesium is basically stabilizing the DNA so that the DNA polymerase enzyme can come along and do its job. Coenzymes, on the other hand, have no catalytic activity. These are just sort of, I like to think of them as handymen. Right? They're just around, they're just helping out, they're playing a supportive role. But they are also necessary for proper enzyme functioning. Right? These are usually vitamins, B1, B6, so on and so forth. They can be NADH. And basically, their differentiating characteristic is that they're not playing an active role in the enzyme's activity. They're sort of supporting the enzyme. So they can be holding some electrons in the case of NADH. They can help to provide any molecules that the enzyme may need, but they're not playing a direct role in the enzyme's activity. Right? So that's how we're going to differentiate cofactors from coenzymes. And then again, cofactors are usually trace metals, and coenzymes are usually vitamin derivatives.